hard transition, right? Now, when we start showing you this that we showed you originally your freshman year, you're like, wow, there's a lot more detail behind this. Just a little, okay? So in science, we always walk this delicate line of giving you enough information for you to understand conceptually, but not giving you so much information that you get lost in all the details, okay? So you see here that when we talk about the splitting of water, I originally only focus on the electrons. Why? Because I want you to understand oxidation reduction. I want you to understand that sunlight is captured in these high energy electrons, and then ultimately that energy from the sun is captured in macromolecules here. I don't talk about ATP and protons here at all. Why? Because it's too much detail. Okay. So then let's see. So then we introduced a little bit more of the detail here, right? Same thing. We're ratcheting up detail. So now you're getting into photosystem 2, electron transport chain, and photosystem 1. Same thing. This is ignoring the protons, right? You're starting to get into, the light is used twice, and the electrons are excited the first time to produce ATP. But as biologists, we cheat and we just draw the arrow as if it's just this magic process that you draw an arrow and ATP comes out, right? So the electrons are excited the first time, and then the energy is used to create a proton gradient, which is then used to create ATP. Well, of course, at the end of the electron transport chain, then you have this low energy electron. So you have to re-excite the electron, and this is actually the electron that ends up in our food. Okay, the high energy electrons that are produced by photosystem 1 are the electrons that end up in our food. Okay, this is the energy that we use to then to transfer the energy to ATP, then to do cellular work. So there's 3 components here to what we call the light dependent reaction. Okay, and that was off of this handout. Where's the handout? So pretty. Where is it? There it is. Okay, so when we see the light dependent reaction here, and we see the production of NADPH and ATP, right? These two are produced in NADPH and ATP. All of these handouts fit together, okay? So these are the light dependent reactions, and there's 3 portions to it: photosystem 2, electron transport chain, and photosystem 1, right? So when you see these 2 purple rectangles here, understand that these protein complexes, it's multiple proteins all joined together, that these protein complexes are actually embedded in the membrane of the thylakoid. You say, what? The thylakoid is a membrane-bound structure inside of the chloroplast. The chloroplast is a membrane-bound organelle inside of the plant cell. Okay, so when we show you these 2 purple rectangles here, these are protein complexes that are located there on the membrane of the thylakoid. Okay, so now we take this thylakoid, looks just like a jelly donut in all honesty, and we're going to zoom in on that thylakoid. So we zoom in on that thylakoid, we get this handout. So this membrane here is the thylakoid membrane, and this membrane is that membrane right there. Okay, I want you totally comfortable as to where we are in the cell. Okay, we then sat down and said, all right, here's the thylakoid membrane, right? So where should we start? Let's go through this, except you're going to give it to me. Where do we start?
Photosystem 2.
Photosystem 2, got it. That means we need a big old arrow here. So photosystem 2. What happens in photosystem 2? What comes in?
Light and water, or H2O.
All right, so we'll go, you said light first. I heard a couple of voices there. That was cool. I'm not going to color coordinate the hydrogen and the oxygen just for ease. All right, so light and water come in. All right, then what? Oxidation, totally agree. So the water then is oxidized. Okay, the oxidation of water then actually releases 3 things. What 3 things?
Protons, electrons, and hydrogen.
Protons, electrons, and
Oxygen?
Oxygen, yeah.
totally agree. Oxygen. Cool. All right. Of course, these are low energy electrons. So that's why Campbell gave you this purple color here. The purple color here. He's doing this intentionally. The purple color here. Okay, he's trying to tie into the right-hand side of your brain. It's nicely done. So the purple here. This is photosystem 2. This is photosystem 2. All right. So photosystem 2 oxidizes water by a process called photolysis. So photolysis is an oxidative process. Ooh, look at us. Okay, so the water is oxidized. Oxygen, I got me saying it. Oxygen is a byproduct. The proton here, that's nice. And then the low energy electrons. All right, so where should we go next?
All right. So the electrons, lower energy electrons are excited, and these electrons are then transported through a series of proteins in the electron transport chain.
Okay, so this here then is the electron transport chain. The electrons are transported. Cool. So by the time the electrons show up over here, they're low energy electrons. All right. So as the electrons are transported, what's the point? What's the point of the electron transport chain? What's the whole point? To produce ATP, I agree, eventually. Okay, that's step 2. What is the first purpose of the electron transport chain? I totally agree. Ultimately, it produces ATP. Yes, that's part 2. What's part 1? What happens here? This big old red arrow.
Pumping protons.
Yep, the protons are pumped and it produces a proton gradient. The thought crossed my mind, it produces. And so what did I start to write? Produces. Proton gradient. So these proton, the technical term is it is translocated. It is pumped into the inner thylakoid space. Technically, this is called the inner thylakoid space. I think it's kind of a wordy term, but okay. The stroma is out here, right? The stroma is out here, so the proton is then pumped from the stroma into the thylakoid, the inner thylakoid space. Fine. So then you get this proton gradient. Well, gradient is a difference, okay? Between you and I, there's an age gradient. There's a shoe size gradient. There's a height gradient. It's just a difference. So when you have a proton gradient, that means you have a difference in the number of protons inside the thylakoid versus outside, okay? And so what this builds up is a high concentration of protons in here, while it is a low concentration of protons out in the stroma. So once you have a lot of protons in the thylakoid, what are the protons induced to do? People will say, what do the protons want to do? Fine. What do the protons do?
They exit the
They diffuse.
thylakoid.
They diffuse, they exit.
Diffuse.
Great, okay? Now, that diffusion of protons has a special name, okay? What do we call that diffusion of protons?
Chemiosmosis.
Chemiosmosis. I would have preferred chemidiffusion, but again, I wasn't on the committee. So they, whoever they are, man, I want to talk to they decided chemiosmosis. Fine. The biggest point that I want to make here is that this is flow. All right. This is pumping here. This proton here is not given a choice. That proton is grabbed from the stroma and forced into the inner thylakoid space. Okay. Against the concentration gradient for protons. That proton is forced. That's why you needed the energy from the electron. Right? So that proton is forced into the inner thylakoid space. Once you have the gradient, now this is easy. This is downhill for the concentration. The protons then flow from high concentration to low across which enzyme? Nice. ATP synthase. ATP synthase is the enzyme that synthesizes ATP. How? Well, I'll show you an animation tomorrow where you will see as the protons flow through this enzyme, it causes the enzyme to physically change shape. This collar actually rotates, ratchets, right? While the head has 3 different protein areas that open and close. It's really cool. And what the head does is physically grabs adenosine diphosphate, ADP, and the inorganic phosphate and forces them together, okay? Physically makes a high energy bond between this phosphate and this ADP and makes ATP. So the whole point of photosystem 2, the electron transport chain, the pumping of protons, the proton gradient, and chemiosmosis, all of this is to produce ATP. For the cell to live on?
No.
What's this ATP for then? Why even make it? Yes, powers the enzymes in the Calvin cycle, which is what we'll talk about in about 3 minutes. Awesome. All right, but now we have, nicely done. Now we have this low energy electron here. Poor little low energy electron. So what happens now?
Light is used once again to excite the electrons.
Light is used again. You said light first, so I'll do light here. Light is used again in photosystem
1.
1. Right? That re-excites this electron. This is now the electron that is going to be stored in macromolecules. This is the high energy electron that cellular respiration uses. This is the high energy electron that you find in food. There is a small transfer here. Then you get the skull protein. Right? Which protein is the skull protein?
NADP reductase.
NADP reductase. That is the enzyme that reduces NADP. So what goes in that box right there?
NADP plus.
Yep. NADP plus. Technically, there's an associated proton. If you really want to be technical, it's that, if you really want to know. Right? All right, so then the high energy electron handed off to this protein, handed off to this enzyme. This enzyme then bonds the high energy electrons to the electron carrier. One electron goes here and makes that neutral. One electron goes here and turns that hydrogen plus an electron, that proton, bonds to an electron to make hydrogen. That hydrogen then bonds to NADP, and that makes NADPH. So now we have the high energy electrons here, and now we have the power for the enzymes. And where do these 2 items go?
To the Calvin cycle.
The Calvin cycle, where the Calvin cycle is going to take carbon dioxide and high energy electrons. Those high energy electrons are going to be bonded to the carbon dioxide, so the CO2 is reduced, and that's how plants make food. Right? The way that you learn this is unfamiliar. I get it. Okay? The way that you handle biochemistry that is unfamiliar is you go through it, and then you go through it, and then you go through it. This is not difficult. I agree. It's unfamiliar, totally. Okay? But don't be intimidated by it. Just because it's new and just because it's unfamiliar doesn't make it difficult. Okay? All right. Any questions on the light dependent reactions before we leave them?
No.
Okay. All right. Ta-da! Calvin cycle. Discovered by a gentleman named Dr. Calvin, 1950s, 1960s. I don't have that date memorized. So, oh man, this is all about counting, is what this really is all about. So, you have a cycle, okay? Now, in the beginning, let me get rid of all this stuff. In the beginning, this, again, can look intimidating. I promise it's not, okay? You're going to take a 5-carbon compound. You're going to add 1 carbon. So 5 carbons plus 1 carbon is 6 carbons. Do you agree that 5 plus 1 is 6? Do you agree with that math? Yep. Well, then they go from 6 to 3. How do you go from 6 to 3? Well, if you have a 6 carbon molecule, what do you do to that 6 carbon molecule to make that a molecule of 3?
Divide it, split
Break
it.
it in half. Okay. This intermediary here is unstable. We even call it unstable intermediary. We don't even give it a name. Okay. So 5 carbons plus 1 carbon makes a chain of 6 carbons, but it's unstable. So those 6 carbons then break into 2 3-carbon molecules, right? Aha! Here's your ATP from the light-dependent reactions. Here's your NADPH from your light-dependent reactions. You now have a molecule that's carrying the high-energy electrons, right? You take one of those molecules out, you take what's left and literally rearrange it. You don't have to worry about the rearrangement at all, okay? In college, if you're a STEM major, specifically a biology major, you'll get into botanical biochemistry. It's biochemistry for plants. And you'll talk about how this shuffling reworks. That's the only time any of you are going to have to worry about that, is if you get into the biochemistry of plants. The animations I show you will literally take these molecules and they go, ta-da, and you go from here to here, right? You don't have to worry about the detail of how you get from here to here. You just have to know what happens, and then the process restarts, okay? So there is this cycling of carbon. Every time that you add 3 carbons on the top, you spit out 3 carbons on the bottom. That's actually how this works. So what we want to do is go through, and this is literally an exercise in counting. So these little black spheres are, where's my highlighter? Around here somewhere, there it is, are carbon, okay? So I'll highlight these. 1, 2, 3, 4, 5. You guys are IBers, you can count. 1, 2, 3, 4, and 5. That's a 5-carbon compound. And you say, all right, and what are these things on the end here? Well, when Campbell's puts these yellow circles on the end, that's always phosphate. So there's a phosphate here, and there's a phosphate there. So there's a phosphate on each end, right? Then there's an enzyme called RUBP carboxylase, okay? So this is RU, that should be a lowercase u. RUBP carboxylase, okay? Well, this is an enzyme that adds carbon dioxide to RUBP. So this 5-carbon molecule right here, what do you think the name of that molecule is? Considering the enzyme that works on it is RUBP carboxylase. What do you think this is called? Yep. So when you see the name of this enzyme and you go, what? RUBP carboxylase, why? Because this is the enzyme that carboxylizes or adds carbon, bonds carbon to RUBP. What is RUBP? That's this 5-carbon compound here, and we call it RUBP. Now, technically, what is it? It's ribulose, which is a sugar, and then there's a phosphate on the 1st and 5th carbon. So it's 1, 5, because it's the 1st carbon has a phosphate, and the 5th carbon has a phosphate. There's 2 phosphates, so it's bi, and there are 2 what? Phosphates. Okay? Now, no one walks around saying, yes, ribulose 1,5 bisphosphate. Nobody says that, okay? What does everybody call this? RUBP. RUBP is simply a 5-carbon compound where the Calvin cycle starts, You have this 5-carbon compound called RUBP. Fine. Then you have an enzyme, RUBP carboxylase. Now, some of your professors might call this enzyme by its nickname, Rubisco. I've never found out why they call it Rubisco, but some technical botanists, they refer to it as Rubisco. So if a professor throws you the carboxylase, fine. If they throw you Rubisco, fine. So then carbon comes in the top. Okay, so this is your 1 carbon coming in. This is the carbon dioxide that we all know plants absorb. We know that plants absorb CO2, yes?
Yeah.
Yes? Okay, cool. However, look at the product on the bottom. How many carbons leave the bottom? How many black spheres do we have?
3.
3. So how many carbon dioxides do you think have to come in the top, considering this is a cycle? If you have 3 out, how many come in?
3.
3. For a total of 3 carbon. Okay, well, hold it. We said 5 plus 1 is 6. But if we have 3 carbon dioxides coming in, how many RUBP do you think are used? If you have 3 carbon dioxides coming in, how many of these do you think are used?
3.
3. If you have 3 of this 5 carbon molecule, how many total carbons do you have here? If you have 3 of these, how many total do you have?
15.
Would it make sense that you have 15 carbons? You start with 15 carbons. Let me zoom in on this. You start with 15 carbons on the top left. 15 plus 3 is going to give us how many carbons over here?
18.
18. 15. Wow. 15 plus 3 is going to give us 18 over here. And you say, hold it. How is it possible to get 18 carbons over here? Well, how many carbons in each of these unstable intermediaries?
6.
6. 4, 5, 6. Then, of course, you have the phosphate. I should highlight those too. You have the phosphate on each end. PO4 here. PO4 here. Always have to get the charge. This is called an unstable intermediate. It's so unstable. Unstable intermediate. It's so unstable, we don't even give it a name. Okay? Well, if each unstable intermediate has 6 carbons, and it was 5 plus 1 is 6, if you had 3 of these, how many of these do you have? If you had 3 of these,
3.
how many of these?
3.
3. And each one has how many?
6 carbons.
6 carbons. So if you have 3 6-carbon molecules, how much do you have total? What's 3 times 6?
18.
You have 18 carbons. Okay. So what Campbell has done here is they've only shown you one. You get RUBP, you bond 1 carbon to it, and that makes a 6-carbon intermediate. Okay. But this 5 plus 1 for 6 happens 3 separate times. So you get 3 RUBPs, and you attach a carbon, a carbon, a carbon, and you end up then with 3 unstable intermediates. So this really, there's 3 of these, right? Each one picks up 1 carbon, and so you get 3 intermediates over here. Does that make sense?
Yes.
You cool? Okay.
Are they
No.
all linked together?
I will show you.
Okay.
Here's it. I drew 4. Let's do this. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. You get 3 RUBPs. Like such. Okay. You then have 3 separate CO2s, which then gives you 1, 2, 3, 4, 5, 6. 4, 5, 6. 1, 2, 3, 4, 5, 6. And you end up then with 6 unstable intermediates. So you get 3 RUBPs times 5 each is 15 carbons. You have 3 carbon dioxides with 1 carbon each is 3 carbons total. Each of these picks up 1 carbon, like such. So here's your addition. So now what you're left with here are 3 molecules of 6 carbons each for a total of 18 carbons. You comfortable there?
Mr. Beno,
Yes.
for the unstable, what's it called?
Intermediate, the unstable intermediate. Mm-hmm.
Yes. Are they all single bonded or is there any double bonds?
They're all. There's some double bonding in there.
Is there anything that decides which are double bonded?
Yes, the type of plant. Plants bond their carbon differently. There's 3 different styles to this bonding. When you get into botany in college, they'll talk about what they call C3 plants, C4 plants, and CAM plants. And so the intermediate here is different depending on the type of plant. But
Okay.
that's way beyond the scope of this course. You're going to get that when you're about 20. That's coming when you take botany in college.
All right.
Same thing. It's not difficult. It's just I'm trying to teach you about soccer, like kick the ball into the net, don't use your hands, and you're asking me about offsides. Does that make sense?
Yeah.
Okay, cool. It's not that you can't handle it, it's just way too early. Okay, so even though this is happening with 3 different molecules at the same time, Campbell only shows you 1. Okay, and this step here of taking carbon and attaching the carbon to this substrate is called carbon fixation. To fix carbon is an old school term of bonding carbon dioxide to another molecule. So you took RUBP, well, you didn't take it, a plant takes RUBP and bonds CO2 to it. That's called carbon fixation or fixing carbon. Okay, all right. So this is unstable. This is a long 6 carbon chain, right? But now we only have a 3 carbon chain. So what do you think happens to that 6 carbon chain?
It splits.
It splits. Okay. So if you have 3 of these molecules and each one splits in half. So now what you're left with is this, this, this. How many 3 carbon compounds are here?
6. Oh, 9.
1, 2, 3, 4, 5, 6. You
Sorry.
agree? No, you're right. Oh, I get it. Okay. So now you have 6 of these molecules. How many carbons each?
3.
3 for a total of how many carbons total?
18.
18. So has the number of carbons changed?
No.
No, there's no change here at all. All you did was you took a 6 carbon chain and cut it in half. That's it. You did that cut 3 different, well, you didn't cut it, they broke apart because they're unstable. These unstable molecules broke apart into these 3 carbon compounds. Okay, so that's what he's showing here. Okay, so again, we have, let's number these. 1, 2, 3. And then, of course, each one has the phosphate on the end, like such, PO4. Okay, but here are your 3 carbons here. 1, 2, 3. Okay. Okay, cool. This has a name. It's called 3-phosphoglycerate, if you want to know. It's called 3-phosphoglycerate. But nobody calls it 3-phosphoglycerate. Why do we call it 3? Because the phosphate is attached to the number 3 carbon. So 3-phosphoglycerate. Nobody calls it that. We call it PGA. We simply call it PGA. And how many of these PGAs do we have?
6.
6 of them. You have 6 PGAs for how many total carbons?
18.
18. All right. And this process then, once the plant produces the PGA, ATP comes in. And when ATP comes in, it donates a phosphate. So that's why you see a phosphate here on the end and a phosphate here on the end. Here, you only had 1. Now you have 2. So now this is no longer just 3-phosphoglycerate. Where are your phosphates? On which carbons?
1 and 3.
1 and 3. And how many phosphates are there?
2.
2. So this is now 1, 3. How do we say that there's 2 phosphates?
By.
Yep. This is phosphoglycerate. Do you think anybody calls it that?
No.
Nope. We call it BPG. So come on, everybody knows that PGA bonds to ATP, releasing ADP, and then producing BPG, right? Okay, this is where science gets really muddy for a lot of people because all they hear is a bunch of letters, okay? So don't be confused by what this is telling you. You have a 3-carbon compound. This 3-carbon compound happens to be called PGA, all right? This 3-carbon compound, PGA, grabs a phosphate from ATP. The PGA grabs a phosphate, and now this PGA has 2 phosphates. Thus, it is biphosphoglycerate. Thus, the BPG, right? Have we lost any carbons here at all?
No.
No. So we still have 6 BPG for a total of 18 carbons. That's it. Now, what was the other product of the light-dependent reactions? ATP and what?
NADPH.
NADPH. Okay. NADPH carries those high-energy electrons. Okay. So those high-energy electrons are going to be transferred to BPG. Well, since NADP is giving up its high-energy electrons, what process am I going to put right on that line right there? Since the electrons are being taken away from NADPH.
Oxidation.
That is oxidation. When NADPH is oxidized, it gives up its electrons and becomes NADP again. Now, if you really want to know, there's an associated proton. And then also what comes out here is one of the phosphates, which I should have written in black. All right. Well, if a molecule is being oxidized, then what has to happen to a different molecule? As NADPH is being oxidized, what happens to BPG? It's reduced. Very nice. BPG is reduced. BPG takes in those high energy electrons. So now this 3-carbon molecule right here has high energy electrons. It still has 3 carbons. It still has the phosphate on the end. Right? The difference here is this has high energy electrons. Okay? This compound is called glyceraldehyde 3-phosphate. Why is it called glyceraldehyde 3-phosphate? Because this 3-carbon compound is a glyceraldehyde, and then the phosphate is on the 3rd carbon. So this is glyceraldehyde 3-phosphate. But do you think anybody calls it glyceraldehyde 3-phosphate?
No.
Nope. What do we call it? G3P. That is actually the product of photosynthesis. That's really what photosynthesis produces, is G3P. From G3P, plants can make any macromolecule. So then what happens is one of the G3P leaves. Oh, I should write that. Will green show up on that if I write that like that? Yeah, that's all right. Okay. So we had 6 PGAs, 6 BPGs, which means here we have, of course, 6 G3Ps. However, 1 leaves. Well, if you have 6 and 1 leaves, how many G3Ps are over here?
5.
5. If you have 5 G3Ps over here, how many total carbon do you have?
15.
15. Right? 15 plus 3, 18. 18, 18, 18. 3 leave. 15, 15, 15. So this side of Calvin is all 15. This side of Calvin is all 18. 3 in the top, 3 out the bottom. Do you see that? Because there's a ton of detail here, okay? Understand, where's my post-it notes? I need a post-it note here. I need a post-it note. Actually, I should do it this way. Here. There's a saying that you have to see the forest for the trees, right? Sometimes you got to step back and see the big picture idea here. Can I zoom that out anymore? Can't. Ha. How many carbons all over here?
15.
15 carbons. How many come in the top?
3.
Therefore, how many carbons are all over here?
18.
18. And then what gets spit out the bottom? How many?
3.
3. And you're back to
15.
15. Okay. The Calvin cycle is really interesting, that you need to be able to step back and see the big picture. Okay. What's the big picture? You start with, or the plant starts with 15 carbons. Okay. 3 carbons come in the top to make 18 carbons total. All right. You then, of course, add the high energy electrons that end up leaving down here, right? It's 18 carbons, high energy electrons in, those high energy electrons come out, and you're left with 15 again. And then add 3 high energy electrons, spit 3 out, back to 15. This is the Calvin cycle. This is what plants do. Now, you can be easily overwhelmed by all the terminology too, okay? So let's do a general summary so that you're also comfortable. So there's 15 carbons over here. You then bring in plus 3 carbons. You then spit out 3 carbons, and that leaves 18 carbons. You add high energy electrons here. you add high energy electrons here. Which molecule is here on the top left? RUBP. Okay. RUBP then bonds to 3 carbons, makes an unstable intermediate. That unstable intermediate breaks down, separates into
PGA.
PGA. You then add a phosphate to that PGA, and that forms
BPG.
BPG. You then add the high energy electrons, and that makes
G3P.
G3P. Yep. One of the G3P then leaves. That leaves you with fewer G3P. And then some chemistry magic happens, and this is reconverted into RUBP. That's how I want you to see the Calvin cycle. I have to give you all the details first, and you're like, "Oh, it's like a bucket of water in the face," and I get that. Okay, you see this, and you're like, "What?" Okay, then you have to back off and say, "Look, there's a molecule RUBP, bonds some carbon, makes PGA. Add a phosphate, BPG. Add high energy electrons, G3P. What comes out? One of the G3Ps. That should be a P, by the way. Okay, and then these are rearranged into that. Okay, so this is something that you need to see 3, 4, 5, 18, 35 times, right? You learn this by just seeing it over and over and over again. So I think that is more than enough content for today. All right, I will see you tomorrow morning for the usual Friday class, and then I will see you a little bit before 1 o'clock. And we will talk to some pharmaceutical researchers for about an hour from AstraZeneca. And then that'll be one more week closer to your graduation.